organisms in Earth are made up of chemicals heavily based on the element carbon. Carbon enters the biosphere through the process of photosynthesis, where plants use solar energy to transform CO2 into molecules of life such as glucose. So now that we know what a carbon is in a basic level, what is it that makes carbon so special? Of all the chemical elements existent, carbon cannot be compared in its ability to form molecules that are large, complex, and diverse. These molecules account for the molecular diversity of life. What makes carbon capable of making these large, complex, diverse molecules lies in the fact that carbon has 4 valence electrons, in other words, tetravalence. What this means is that carbon would have to donate or accept 4 electrons to fill its valence shell. But instead of forming an anionic bond, carbon could covalently bond to other atoms and share its 4 electrons. Let's take a look at this in an electron configuration diagram. This is an example of how carbon covalently bond with other elements. Look at this. You have 4 valence electrons. On the other end here, we have only 1 valence electron. Carbon can make 4 covalent bonds with 4 different hydrogens. In the end, we get the product of CH4. These bonds that carbon forms with other elements can either be single bond or be double bond. This particular electron configuration is what allows carbon its compatibility to form covalent bonds with many other elements such as hydrogen, oxygen, and nitrogen. How does cells synthesize and break down products? The second topic we'll discuss today is regarding the 2 processes named dehydration synthesis and hydrolysis. If you look at the structure of a carbohydrate, protein, and nucleic acids, you notice that they're all polymers. This means that it's crucial for cells to be able to synthesize monomers to form polymers, and on the other hand, to be able to decompose polymers to form monomers. The process of synthesizing monomers to form polymers with the loss of water is called dehydration synthesis. When a bond forms between 2 monomers, each monomer contributes a part of the water molecule that is lost. This monomer is contributing an hydroxyl group, and this monomer is contributing an hydrogen. And when water is lost, these 2 monomers form a new bond, making a polymer. The reverse reaction of dehydration synthesis is hydrolysis. Just like its name, where hydro means water and lysis means break, it adds water to break polymers into independent monomers. When water is added, one monomer gets the hydrogen and the other monomer gets the hydroxyl group, separating the polymer into independent monomers. An example of hydrolysis and dehydration synthesis is the process of digestion. The food we absorb is usually in the form of polymers, making it hard to enter our cells. This is where enzyme comes in to speed up the process of hydrolysis. By the end of hydrolysis, these molecules that are too large are broken down and they end up in the bloodstream where they're eventually distributed to body cells. Then, these cells use dehydration synthesis to form polymers with specific functions by the use of monomers. How does the structures of biologically important molecules account for their function? Carbohydrates are basically categorized into 3 groups: monosaccharide, disaccharide, and polysaccharide. All these groups differ in structures, thus functions. Monosaccharides are simple sugars. Because they are basic monomers of carbohydrates, their main function is providing energy like a fuel and later being assembled into disaccharide or polysaccharide. For the case of disaccharide, where there are 2 monosaccharides, their function becomes a little more complicated. Sucrose, an example of a disaccharide, allows plants to transport carbohydrates from leaves to roots and other photosynthetic organs. When it comes to reach the stage of a polysaccharide, many more complex functions arise. These functions include providing structural support regarding the cell wall and storing energy through polysaccharides such as glycogen. From this, we can see the functions become more complex as the structure becomes more complex. There are three types of lipids: fats, phospholipids, and steroids. Out of these three, phospholipid structures directly correlates with its function. First, let's look at the structure of phospholipids. The phospholipids are consistent of glycerol attached to two fatty acids. The glycerol part is the head, whereas the two fatty acid chain is referred as the tails. The heads are hydrophilic side, and the tail is the hydrophobic side. These phospholipids are components of the cell membrane that regulates what goes in and out of the cell. Because phospholipids have a polar and a nonpolar side, it wouldn't accept overly charged molecules to come in or out of the cell. This contribution of phospholipids accounts for how the cell maintains a stable environment using its cell membrane. Proteins out of these four molecules definitely has much more structures in comparison. With four levels of structure: primary, secondary, tertiary, quaternary. It has diverse functions as well. In most of the cases, functional proteins depend on its ability to recognize and bind to some other molecule. This also means that if factors like pH came to denature the structure of protein, it will lose its function. A representative example of this is an enzyme. An enzyme and a substrate are extremely specific in its shape. An enzyme for a certain substrate rarely interact with other types of substrate. That is why when an inhibitor comes and changes the structure of the enzyme, the substrate is no longer able to bind to an enzyme and is unable to carry out the chemical process. There are two types of nucleic acids: RNA and DNA. Both of these nucleic acids have intimate relationship between its structure and function, but today we'll be looking at DNA specifically. The structure of a DNA is a double helix. This structure is a perfect match for the function of DNA, specifically the process of replication. Because DNA is a double helix, when the time for replication comes, one old strand can be used as a template for the new forming strand following the base pairing rule. If it were any other structure, it would have been harder or even impossible to produce the exact copy of parental genes for the offspring. Through this video essay, we have learned, first, how carbon's 4-valence electron is responsible for the molecular diversity of life. Second, the process of hydrolysis and dehydration synthesis. And lastly, we have learned how the important biological molecules' structures relate to their functions. Thank you for watching, and I'll see you next time.